Okay, so it is Wednesday, and it is what, June 4th. Yeah, June 4th. And we're going to finish up the last little bit of your chapter 3, last little bit about the transcription, translation, genetics, mutation story. We'll then be moving into a chapter on histology. Histology is something you've been exposed to in lab, and so I'll run through that and give you some highlights and hopefully some reminders. And then that will leave for us next Monday a conversation about skin. And then next Wednesday, a week from today, we have an exam. Okay, and that'll be exam number 2. It'll be a full-length exam, and you'll need a Scantron form. You'll have the entire period to do that, and it will cover all things about the cell, Chapter 3, Chapter 4, histology, and Chapter 5 on the skin. Vocabulary on that exam, 21 through 40. And I'll go through 27 through, about 34, 35 today. And then the last few slides next Monday. So if you haven't already, go ahead and make up those note cards for 21 through 40 so that you're not playing catch-up on the vocab next week. Any questions regarding vocabulary? Everyone feeling comfortable how to proceed with this? Okay, let's go through the vocab, then I'll give a little recap of the material up through where we are in the lecture. So we already did this one last time, and moving on, if you see the word organelle, right? And another way of thinking about an organelle, it is a small organ, right? So L means small. So the organelles. Now I know a couple of you were struggling on the last exam, and you had organelle in the wrong place of the hierarchy, and you were confusing organelle with organ. Okay, so maybe this will clarify that if that was a confusion for you. But the organelles are the building blocks, the Golgi, the mitochondria type structures within the cell. M and N both mean in or into. Embol, a stopper. So an embolism is literally a blood clot that goes to the lung, a stopper of the blood. And then emesis is vomiting. If you had dysemesis, what would that mean? Dys was good, bad or difficult, right? So dysemesis, bad, vomiting, like there's good vomiting, but bad vomiting, or hematoemesis would be vomiting of blood. Emia is any kind of condition of the blood. So if you see, for example, anemia, you're lacking something important in the blood, a reduction in the red blood cells or in the iron levels. But if you see emia of any sort, it's something to do with the blood. Encephal, or encephalo, is the brain. What did cephal mean? Cephal meant head. Encephal, within the head, one finds the brain. Okay, encephalitis, an inflammation of the brain. Endo, within. Entero, your intestines. So enteritis, inflammation of the intestines. Epi, on or upon. The epidermis is on the dermis. In lab next Monday, we'll be discussing the epiphysis, the epiphysis, which is on the growth plate. So you'll see epi in a few places. Erythro, red. Erythrocytes are your red blood cells. Estha is sensation. Anesthesia, a lack of sensation, a negating term. And then EU, good. Now next to this you can also put well or even true as other meanings of EU. You'll hear it said that we are eukaryotic organisms because we have cells with a true nucleus. If you give a eulogy, you give a good word, right? So we see this EU turn, or eupnea would be good or normal breathing. Ex and exo, both outside. To exhale, to exit, exocytosis, to move things out. And extra also on the outside. So extracellular, something outside of the cells. Fasci, a band sort of structure. We'll see fascicles later in the course. Fascicles are bands of structures in muscle and nerves. And then fever, or FEBR, fever. So if you're febrile, you have an infection which is causing a fever. Ferent, to carry. Efferent, afferent. We'll see those words later on. We saw the AF meant toward, EF meant away. So efferent or afferent, something's being carried away or being carried toward. Fibrofibers. What cells make those fibers? We've seen these cells in lab. Fibroblasts. So the fibroblasts were the cells that were forming or making the fibers. Fizz is a split, or even you could change it to slit, because what we're talking about is something that divides. A fissure is a small, narrow opening or slit or split between bones. Flux, flow, reflux, right? To taste again, to have backflow, if you will. Foramen is an opening in a bone. A lot of that in lab next Monday for most of you. If a term ends in form, it's usually resembling or in the shape of something. So fusiform is in the shape of, and that term may not make sense to you, but fusiform is a spindly shaped cell. We'll see those when we get to smooth muscle. Fove is a pit or depression. In your eye, there's a structure called the fovea centralis. It's literally a little pit, a little groove in the back layer of the eye. Within gamo, marriage or union, you see the term monogamy, right? Single union, or gamete. Remember, the gametes are the sex cells that unite upon fertilization. Ganglio is a ganglion. That doesn't make sense to you yet. It will when we get to the nervous system. Gastro, the stomach. A gastric ulcer is a stomach ulcer. If you see genesis in a term, it means that it's producing or originating something. We saw the term angiogenesis last time. That is, that tumors are good at producing or encouraging angiogenesis, the formation of new angios, new blood vessels. Genic, same thing, to produce. So carcinogenic, something that's carcinogenic is something that causes cancer. Or, as we'll see, pyogenic bacteria. Pyo is pus. So a pyogenic bacterium is one that is producing pus. I'll go through one more for us today. Gero. Geriatrics, dealing with age, or progeria. You may have heard of this condition where these children are dying of old age by their teenage years. So progeria, and a rapid aging process. Gest, to carry or bear. Think gestation, the period of pregnancy. Gingivo, I've got my examples here flipped, but gingivo, you know, the gingivitis is inflammation of your gums. So gingivo is your gums. And glia means glue. We'll see some glial cells later. Sometimes they're called the neuroglia cells. And these are the cells that basically hold or glue the neurons together in the nervous system. So that's quite a few vocab terms, and we're going to continue through 21 through 40. So on Monday, I'll only have to go through histero, okay, the last few. Chapter 4. I'm going to flash through this quickly and get up to where we were. Just as a quick review, if there's something that I need to restate, let me know. But we talked about the overall structure of DNA as being a chromosome. We discussed the cell cycle and the fact that cells are growing very, very quickly. Some are, and that they're completing their cell cycle very quickly. Others do so more slowly. And we appreciated how important the cell cycle is in the early development, because if the embryo does not rapidly grow, then it won't have a chance of embedding into the uterus. We mentioned the cell division is the process of mitosis, and we also can now recognize the different stages of mitosis. We also discussed briefly the formation of sex cells, that is the gametes, and that requires not mitosis, but instead another type of cell division called meiosis. And we know that meiosis, things can go wrong in meiosis, for example, and form a non-disjunction where the chromosomes don't separate properly during this process, and can result in Down syndrome or trisomy 21. We know that cells undergo apoptosis. They undergo programmed cell death. They die when they're supposed to. They know when to divide. They know when not to divide. There's a tight regulation over this thing called the cell cycle. And tumors are cells which have lost that tight regulation and are growing now out of control. And we described the different characteristics of tumor cells. That is that they They kind of become de-differentiated. They become more simplistic in their identity. They grow rapidly. They have the tendency to encourage angiogenesis, new blood vessels. They lose that idea of contact inhibition, so they tend to crawl on top of each other rather than stopping when they touch each other. And we describe the difference between a benign versus a metastatic cancer, and that is metastatic cancers, in addition to all of those things, tend to travel to places where they normally wouldn't be. Reminder that on exam 2, there will be a short answer section, and I described that to you. You'll be writing 3 sentences, no more than 3 sentences, using 10 of those 11 terms. We then went into DNA and RNA, a little bit more about the structural differences in the two. We described how purines match with pyrimidines. That is, C's always match with G's and A's always match with T's. And this complementary arrangement is very important so that the cell can replicate or copy the DNA very, very quickly. It also gives DNA a very standardized size, so that one purine is matching up with one pyrimidine. We discussed how DNA is copied, that is, it's replicated during the S phase of the cell cycle, and that replication is a very rapid process. In our cells, somewhere around 50 nucleotides per second. So your cells are chinging along in CGATAA, and they're copying at 50 nucleotides per second. We have to go fast, right? Or else our cells would never be able to divide. We've got billions of nucleotides to copy in every cell to replicate our DNA. So errors do occur. And those errors occur at a rate of about 1 in every 100,000 nucleotides, which is pretty good, except we have billions of bases. And so every time our cells do divide, there are going to be small changes or mutations made in our DNA. And that DNA, those changes in the DNA can accumulate to the point where they can become a problem. Those mutations can arise through errors in replication because it's going so fast, but also from environmental exposure to carcinogens, to UV radiation, to X-rays. Even oxygen can cause mutations. So breathing is a health hazard. Okay. We also described how a cell has to accumulate multiple mutations for it to become cancerous. And some of us are born with more of these mutations. We have a family history of a certain kind of cancer. So we're already born with more of these mutations in our cells, making us more likely to get cancer at a younger age, while others don't have that family history. So for them to get this cancer, they would have to accumulate those mutations in their lifespan. And where we're heading with this is that if we make changes to the DNA, then those changes will also be transcribed in the mRNA. And then those mRNA changes would then be also translated into protein, such that it's possible that the cell is now making a mutated protein, and a mutant protein probably doesn't do its job as well. And so that's kind of where we're heading with this, but I want us to understand just a little bit more about the process of transcription and translation. How is it that the cell takes its DNA code sequence that's on the chromosomes and a gene, and how does it get that information out to the ribosomes where proteins are made? And this is this double process of transcription followed by translation. Describe to you that our genome is much more limited than our proteome. That is, we have fewer genes and we do proteins, and the way that the cell makes up the difference is that RNA undergoes processing, and that many pieces of junk are cut out. The exons are kept, the introns are clicked out, and the cell also has the ability of rearranging these pieces so that one gene can actually code for multiple proteins. We looked at the overall process of transcription and translation, and here's a nice overview of that. So in the cell. Here's the nucleus. I know it's the nucleus because I see these pores, right? There's these big gaping holes in the nuclear membrane. And I see the DNA, very simplified here, but I see a little bit of double-stranded DNA in the nucleus. And I see that this single-stranded mRNA is being made. And that first step would be called transcription. And that transcription process has to occur in the nucleus because that's where the DNA is found in a cell, in a eukaryotic cell, in our cells. And then that mRNA is going to go out of the nucleus. It's going to go through those nuclear pores out to the cytoplasm. And in the cytoplasm, what will it run into? That mRNA will run into either ribosomes kind of freely floating around or ribosomes that are attached to the rough ER in that big series of membranes that are wrapped around the nucleus. So the mRNA basically becomes captured by the ER. And there, the ribosomes will work to translate that message into protein. So in this picture, those little green cubes are amino acids, and those amino acids are linked together to make a protein. And these little, looks like little, oh, what's that game with little ball? Jack. Looks like little jacks, right? In that picture, they look like little jacks. Those are the tRNA molecules. And we were finishing up with that conversation last time. The tRNAs are the transfer RNA molecules, and they're going to transfer what? Carry or transfer amino acids. They're going to bring the amino acids in one at a time, and we'll see where that code is here as we finish this up in the next couple minutes. Okay, so I think we're about where we were when we finished up. And I was at this slide. Let's pick up right here. And this is showing you the big picture of things. That is, on the top, in red, this is DNA. I know it's DNA because it tells me it is, but also because I see thymines. I see T's in that sequence. Now, this is only one of the two sequences. The other strand has been removed for simplicity here. That DNA will undergo the process of transcription. Again, this happens in the nucleus, and we make mRNA. The process here uses the same idea of complementation. That is, when the DNA has a C in it, it'll form an mRNA that has a G in it as a complementation. And if the DNA has a T in it, it'll match it up with an A, make an A. And if there's an A up here, though, in the DNA, it will put a U in. Remember, RNA does not have T's, it instead has U's. So when you see U's in a sequence, you know that it is an mRNA, or at least an RNA molecule. And then we're going to see how does the cell take this messenger RNA and from it translate it into protein. So reminder, what is a protein? A protein is a long chain of amino acids. And how many different amino acids are there? 20. So there are 20 different letters in this alphabet, and every protein has a different arrangement of these amino acids. If we look at this green chain, we see that each box, each rectangle is a different amino acid. So you're not responsible for the names here, but for example, cis, seer, lu, tier, glin, these are all amino acids. And they're linked together to make this long thing called a protein. And then what the protein does is it winds up, right? It winds up into this arrangement. And that is the final three-dimensional structure of the protein. Does every protein have all 20 amino acids? No. So some proteins would have more of one than the other. They don't all have to have every amino acid or have a representation of each. So different proteins have a different collection of them. Now, what the amino acids do, though, is they all have their own shape. And that brings us back. Let me go back. The idea that every protein has a specific shape brings us back to what idea? What did we talk about in Chapter 2? Proteins had a particular shape, and we talked about that lock and key idea of an enzyme. So enzymes are proteins that have a particular shape. And if that shape gets changed, then the protein, the enzyme, can no longer do its job. Same idea here. So if we have, thinking ahead, if we have a mutation in the DNA, change in the DNA, it can change the amino acids. Now the protein would not have the right shape. And the shape of the protein is critical to doing its job. So if we start monkeying around with the shape of the protein, we will lose the protein's function. Typically, we lose. Darwinian evolution tells us that sometimes those changes are advantageous, and so that there are positive changes that can occur, potentially, through these mutations. Also, let's keep these mRNAs in order. And my list on the left and the table I know are a little bit out of sync. But we've talked about, in passing, we've now talked about 3 different kinds of RNA molecules. They're all RNA, right? They're all made up of A, C, Gs, and Us. But they have different jobs, different forms. There's messenger RNA. That's the one we've been talking about that goes from DNA to mRNA to protein, right? That's messenger RNA. In this picture, it's shown in yellow. I think in all of our pictures, it's been a yellow single-stranded molecule. But there's also ribosomal RNA. I mentioned this molecule in passing because ribosomal RNA is important in making ribosomes, as the name suggests. And remind me, where is ribosomal RNA made? Where in the cell is ribosomal RNA made? Am I hearing the nucleolus? Right, the nucleolus, that dark center within the nucleus. The nucleolus is where we're making the RNA, the ribosomal RNA. And ribosomal RNA is important for making ribosomes. Then finally, transfer RNA, and we've seen these already. They look like little lunar landing molecules, and this is a tRNA right here. In the previous picture, they look like little jacks. And those are the molecules that are going to be carrying the amino acid to the ribosome as needed. So I alluded to this last time. If you're still a little bit confused or still not getting this story straight, remember that transcription does alphabetically come before translation. And I've already alluded to this idea that transcription is the copying of something, right? If you go home and you transcribe your notes, or in the old days, there were transcriptionists, right? What did Lucy, right? Lucille Ball, in all of her little get-ups in her shows. Lucille had a boss, right? She'd go in and she would transcribe. I have this episode in my mind, right, where she goes in and she's taking shorthand or transcribing the notes. So she was doing her best to copy it, right? But she's still in the same language. So she's going from DNA to RNA or from, transcribing. And then the ribosome does the translation work, and that is changing it completely from a nucleic acid to an amino acid. Remember that there are only 4, right, different nucleotides, A, C, G, and U for RNA, which then are translated into proteins which are made up of 20 different amino acids. So there's some sort of translation that's going on here from one code to the next. Now, one of the things that Watson and Crick described, in addition to building their little model of DNA accurately, was they said, if this is what DNA looks like, then it must be that DNA makes RNA and RNA makes protein. That was that idea of the, I'm going blank now, the central dogma. Right? I mentioned the central dogma, the way things must work. And they also proposed correctly, hypothesized correctly, that the genetic code, all of this must work on a system of 3s. We won't get into the mathematics of this, but if the cell worked on a system of 2 or worked on a system of 4, it would not work. So they hypothesized correctly that the genetic code is working on what's called a triplet, or on a 3-letter code, or 3 bases. And that is also referred to as a codon. It's the coding, right? It's the coding portion of this. So don't worry about the math down here, but just realize that the genetic code is based on series of 3s. This was the 50s, right, when they described the DNA structure. And during the 60s, scientists discovered or created this table. And this is basically the translation table. This is the code that they had to figure out, how do cells understand how to make proteins? And what they realized is that those 3 letters and those 4 nucleotides making 20 amino acids had to have a connection as such. This table can be called the genetic code. So I told you that the word genetic code, which is one of your 10 words to put into sentences, can be used in 2 different ways. You'll hear it said that your cells contain the genetic code, right? That our DNA contains the genetic code. And then others will say that this table, this translation table, is also, quote, the genetic code. So you can use that word in a couple of different ways. Here's what the deal is. You've got a ribosome. The ribosome is receiving what molecule? The ribosome is receiving the mRNA. And the mRNA is bringing message in from the DNA. And the ribosome is looking at things in 3-letter sequences. So every time the ribosome sees, for example, every time the ribosome sees CCC in a row, it knows that it's supposed to translate that into this amino acid, proline. And every time the ribosome sees ACU, it instead will translate that into threonine. Okay? So this is that code, and we found out that this code is used for every organism on the planet with just a couple of exceptions. So this is true for bacteria, this is true for yeast, this is true for plants, for animals, for you and everyone else. So every plant, every biological system on the planet uses the same genetic code. So let me show you how to use this. Now, it turns out there are 3 of these letters that basically act as a sentence. They tell the ribosome that the protein's done. We call these stop codons. They're listed here. So when the ribosome sees UGA, it knows basically that, hey, I'm done. And the ribosome dissociates, falls apart, and that protein is now complete. So again, we see this picture, and we realize now, looking back at it, that it's all in groups of 3. So the DNA, we break it up into groups of 3. Each of these is called a codon, and the ribosome is able to look at each of those groups of 3, and using that code, that table that I just showed you, determine what amino acid should be there. So if we go back and look, I assure you that CGU, these 3 letters, let's look back. CGU is going to make this protein ARG. Let's go back and find CGU. First letter C, second letter G, there it is, CGU. And every time the ribosome sees CGU, it knows to put in arginine. Okay, so we see that this thing works, and then again, every time the ribosome sees UCA, I assure you the code will tell us that it's going to make a serine. And every time it sees UGG, it's going to be trip. Okay? So you'll learn how to use this table in the next couple minutes. Now the genetic code is said to be the genetic code is redundant, but it is not ambiguous. Let's make sure we understand what those 2 words mean. If someone says to you, man, you're redundant, what are they telling you? You're saying the same thing over and over and over. And sure enough, the genetic code is redundant, because if we go back and look, we'll see that sometimes the same 3 different letters can give us the same amino acid. So there's some redundancy to the code. But the code is not ambiguous, meaning that every single time the ribosome sees CCC, it'll put in the same amino acid. There's no ambiguity about it. There's no confusion. When the ribosome sees those 3 letters, it will always, always, always put in the correct amino acid. So here's another version of the same table. Let me have you practice for a moment. So which amino acid would the ribosome put in if it saw UUG? UUG, up here, right? And it's called LEU. That's all you need to know. It's leucine, but it's LEU for all you care about. Then, what about for CGG? Find that one. CGG is where? Over here. And again, the ribosome would know to put in arginine. Now, who brought that amino acid over to the ribosome? What molecule brought the correct amino acid over? That tRNA. Right? The messenger RNA had the 3-letter code. The tRNA is going to help bring in the right amino acid, and the ribosome is going to do the translation work. So let's figure out how that works. It's also true that the genetic code is nearly universal. Now, there are a couple of exceptions to organisms that use this genetic code. They are rare exceptions. So I can't say it's universal. Right? It's not absolutely every organism uses the same code, but it is 99.9% universal, which means that I can take a gene from a firefly and put it into a plant, and now the plant will glow, right? And it's why we can take a heat-resistant gene from a tomato plant and put it into rice. And now that rice has the same heat resistance that the tomatoes had, and this is the basis of genetic engineering. So we can take a gene from one organism and put it into another, and because the organism's machinery doesn't know the difference, is that a gene from a bacterium or is that a gene from a human, it just goes ahead and makes the protein. It's kind of a dumb FIFO, first in, first out kind of system. A little bit more complicated, but not by much. So this is why we can have glowing kitties, right? So this little kitty has been genetically modified to have the same gene that this tobacco plant has. And we can express the same gene that's in a firefly. So the same luciferase, the same gene that gives off light, can be activated in the plant or activated in the cat. And now they have glowing puppies and glowing mice and glowing kitties. And you can go on to Google and type in glowing kitties, and you'll see them. And they discovered that the hair gets in the way. So now they have those naked kitties, those hairless kitties. And they've made them glowing, so now they just purr, and they're like little nightlights, right? And the hair doesn't get in the way. Right? They're kind of hideous during the day, but at night they're kind of cute, right? They kind of glow, and they purr, and they keep you warm on your pillow. Why do we do it? Because we can. Should we be doing it? No. Can we have glowing monkeys? Already been done. So what does that mean? Tomorrow could be us, right? Now, I'm not sure we want glowing kids. When you go down to Meijer and you buy fish, and you see those really cool glowfish, there's no fish on the planet that has that neon color like that, those little goldfish with the purple and green and blue and lime green. Those are all genetically modified organisms as well. Okay. And most of the food you're eating is genetically modified. Right? In fact, there's legislation, has it passed or is it still in the talk, that all food labels will have to soon disclose if the food is genetically modified. And what that basically means is that Kellogg's, right, making their corn flakes, has planted corn that has been genetically modified so that that corn is pest resistant or drought resistant or yields a higher yield, right? More corn per acre. So the corn has been genetically modified. We benefit from that, right? Because we have more food on the planet to feed our people, and there's some concern that that is somehow harmful to us. That data is out there and is very, very controversial, but there's a big push right now that all companies will have to disclose if the food, if the corn, if the rice, if the wheat, if the meat that they're using has been genetically modified to increase yield. So that's coming down the pike if it hasn't already made it. Okay, so we've got the ribosome. It reads the mRNA message, and that mRNA message came from the DNA. There's one little piece that's missing here, and that is, how does the ribosome know where to start reading the messenger RNA? It's like when you write your sentences for me next week, how will I know to start reading a sentence? Well, in first grade, we learned to put a capital letter at the beginning. So the capital letter indicates the beginning of a sentence, and then we learned that at the end of a sentence, we put a period. So the ribosome needs the same sort of guidelines, and we call this the reading frame. So there will always be a place where the ribosome starts so that this message will make sense. Let me demonstrate this for you. So if I had, for example, a phrase that went like this: the big red dog. Okay? The big red dog. I'm using three-letter words because we've got a three-letter codon sequence here. Now, the ribosome will know where to, well, first of all, I will know where to start reading that sentence because I see the capital T. So that's my signal to start. The ribosome also has its own little tricks, so it knows where to start. That's outside the scope of this course. Just know that the ribosome knows where to start. And then the ribosome only works in groups of three. So it will have no problem, neither would you, reading this sentence. The big red dog. And then the period, remember I told you there are stop codons, so there'd be another three-letter sequence that would tell the ribosome, hey, we're done, that's the period. But what if there's a mutation? What if there's a change? And what if I lose this letter B? What does the ribosome now read? The next three letters. Garbage, right? So this sentence is no longer useful to me to read, and this protein, right, because we recognize that those three letters represent a different amino acid, right? A different amino acid was put into the protein. This protein is now useless. It's a garbage protein. It has no function. If this was an important protein, the cell no longer can make it, and that cell may very well die. So that mutation could be a lethal mutation. And all that changed was one little letter, right? That B is gone. So that's one way of thinking about how these mutations can have a detrimental effect on your gene. Now, I've already mentioned the tRNA, just to get this whole story and all the players back into scope. The tRNAs were these little jacks, or these little lunar landing molecules, and they were... Oh, I'm late. And they are the ones carrying in the amino acids. The amino acids are these little squares, right? So this amino acid is bringing in Lu. Now, on the other end of the tRNA, see these three little prongs down here? Those three little prongs, remember, this is the tRNA molecule. So what is a tRNA molecule made up of? It's not a trick question. It's RNA. So it's A, C, Gs, and Us. And these three nucleotides right here are called the anticodon. What do we call the three letters here on the mRNA? The codon, right? So what we're going to see is that the codon, the three letters in the mRNA, are going to match up perfectly through complementation the three letters on the tRNA. And this is the true translation mystery right here. Because this is how the mRNA and the ribosome are able to translate that message. When it sees three letters, the correct tRNA comes in, and every tRNA is different, and it carries with it the correct amino acid into the ribosome, and now we continue to build that protein one at a time. So here's just a better view of the tRNA. There it is. There's that anticodon. That anticodon is part of the tRNA, and it binds specifically to the three letters on the mRNA called the codon. Look at how they're matched up. The mRNA says CGA. The C will complement with G. The G with C, and the A with U. That's the connection that's being made. So every time that the cell sees CGA, the ribosome, along with the tRNA, will bring in this particular amino acid, and I assure you that amino acid will be arginine. So again, every time the mRNA carries CGA, it'll match up with a complementary anticodon sequence on the tRNA, which will always bring in the correct amino acid. So that's where the decoding sequence actually occurs. The ribosome, two parts, big and small. Thankfully, they didn't come with anything fancier names than that. So we have a large subunit and a small subunit, which come together to make the RNA. Don't worry about the different sites down here. We won't worry about the EPA stuff. but just recognize that the ribosome kind of slides down along the messenger RNA and one by one matches up so that the correct amino acid is brought into place. So what I've been saying all along is this. If there is a single mutation in the DNA, it can have an effect on the protein, can't it? Even a single little point, a point mutation, can, for example, take out the letter B in the word big and give us junk. Now, some mutations affect the whole chromosome, like I mentioned before, like a Down syndrome situation. That's a different kind of mutation. But what I'm talking about here are these point mutations where maybe there's a insertion of one letter, or maybe there's a deletion of one letter, or maybe the letter is switched, right? Maybe there's a substitution. An A is switched for a T in the DNA, or a C is switched for a G.
An amino acid for every combination? Is there
Yes.
every combination of the...
Yes.
So if it just skipped, wouldn't it just make up a new combination? And just like...
Let's take a look. Hold on. Let's take a look at this example, and I think your answer will become very clear to you. So at the very top, this represents the mRNA. I know it's mRNA because it tells me. I also know it's mRNA because I see what? I see U's, right? So what's missing would be the DNA sequence, right, is above this. It's not part of this conversation right now. And this is the normal sequence across the top. And again, if you go back and look at the genetic code translation page, every time the cell sees AUG, it'll put in this amino acid methionine. And every time it sees AAG, it'll put in lysine. Okay? And every time it sees UUU, it'll put phenylalanine in. Then at some point, UAA, go back and look at it. The UAA is one of the 3 stop codons. And so that's like the period, and the protein is stopped, right? No more. The ribosome knows it's done. Now, what happens in a point mutation where I lose a letter? Like that B in that big word. And if I lose this letter U, then the ribosome is just a dumb translator. It's like Google Translator. You put junk in, you get junk out. And so everything's fine. I have the first 3 letters, right? The ribosome knew where to start. This idea of reading frame, it's staying in the right group of 3s. And then the next 3 letters were the same. But then the U is missing, so now it doesn't see UUU. Now it sees UUG. And sure enough, UUG will be leucine. And then GCU, which will be alanine. And, oh, we just screwed up the stop codon. So the ribosome doesn't know to put a period. It's like some of your run-on sentences that I'll read next week, right? And I'll just keep on going and going and going. And that's exactly what the ribosome does. And all that extra stuff is junk. It's not meaningful to the cell. So you're wasting energy. You're putting together this protein which no longer has any meaning to the cell. Or, you know, sometimes when you're writing an English paper, you put a lot of work into it and the professor goes, yuck. Well, I did a lot of work. Yeah, but it's horrible. It's sort of what's happening here, right? It's not a good combination. Now, what if instead of losing a letter, I add a letter? Same idea, right? The ribosome only can see the next 3 letters in a row. So if I stick in an extra letter, this is especially devastating because now the ribosome sees UAA immediately. Well, UAA is one of those stop codons. This protein isn't even made. It's like, it's one amino acid, done. The ribosome, it's like a one-word sentence, right? It doesn't even get started. So that's also meaningless to the cell. How about a mutation where I don't lose a single letter, but I lose 3 letters in a row? So rather than the big red dog, it just says the big dog, right? I'm losing an entire 3 letters. That sentence still makes sense. It doesn't have as much detail. I don't know that the dog is big, but I still know that it's a red dog. So sometimes when you lose a group of 3, the protein is still made, and the protein might be somewhat useful to the cell, but it may not be fully functional. And then look at this one. Look at this one. A C is changed to a U. Now let's look at what the cell does with that information. The ribosome reads along and it says CC or GGC, and it looks at the chart and says, oh, that's a glycine. And then it looks at again and it says GGU, and the table still says it's a glycine. This is that redundancy thing that I mentioned. The genetic code is redundant. In other words, there's more than one way to say glycine. I can say glycine by GGC, or I can say glycine by GGU. Did a mutation occur?
Yeah.
Was there a change in my DNA? Yeah. But was there a change in my protein? No. So there was a mutation, and I don't have that word on here, but that is oftentimes called a silent mutation. And the reason it's silent, right, it didn't have an effect. Yes, there was a mutation, but it was silent. It had no impact on the organism or on the protein.
Are those the carriers then? Like if you had a mutation?
No, that protein's still the same. That protein is exactly the same. The cell does not know the difference. So in this situation, there's no difference. So those would be just different variations on the same thing.
What would be someone who would carry
A carrier is a person who has a gene that is defective, but when there's only one copy of it, it doesn't cause disease.
okay, so you have
But
to carry.
if you had two copies of it, then you have the disease. So a carrier in genetics is a person who carries one of the genes, but themselves are normal. Their spouse carries one bad gene, but is normal, but their kids, by bad luck, get both of those mutations, and that can cause a disease. And there are a lot of diseases like that, which are silent until two people get together who happen to carry the same genetic mutation. So, another example of the same thing. This is a substitution. Again, rather than losing or gaining, a G was switched into a. Okay. So look at those little examples. And here's one example, one disease that kind of fits into what you were saying. This is sickle cell anemia, just as an example of a mutation that perhaps we've all heard of. Sickle cell anemia is a genetic disease involving the blood, specifically involving a protein called hemoglobin. Hemoglobin is the protein that carries the oxygen through the blood. It's found on the red blood cells. This is the normal sequence of hemoglobin. So what we call wild type, another way of saying normal. Okay, so the normal hemoglobin has in it a CTT sequence, somewhere in the gene for this hemoglobin protein. That means that the mRNA would be GAA, do you agree? Complementary pairing. And GAA on the ribosome is going to give me glue each and every time. In sickle cell anemia, the most common form of this disease, rather than CTT, there's a single T. And within that protein, that T becomes an A. Well, we know what happens. Now, that individual doesn't make the same protein. It now makes a protein that has, instead of glue, it has val in it. Well, that valine behaves very differently than the glue. And as a result, the protein is a slightly different shape. And because the protein's a different shape, it can no longer do its normal job. As a result, these individuals, under times of stress, their red blood cells become sickled in their shape. Now, these red blood cells can no longer travel through the blood vessels without clogging up the blood flow. Now, we know what a normal red blood cell looks like. We've seen them in lab. So up here is a normal red blood cell. I mentioned in lab in passing that they have a very unique size and shape to them. They're about 7.5 microns across. It's pretty tight for a red blood cell to go through your capillaries, the small blood vessels in your circulatory system. And when these cells change their shape, this long sickled shape in the bottom, there's no way that's going to fit through your little capillaries. So as a result, you get a blood clot. Different than a blood clot, but you get a blockage of the blood, is the way I should say it. So these individuals have these episodes where their cells become sickled. Now, what causes their cells to become sickled is this. Hemoglobin normally sits, I'm just going to draw dots, all over or within the red blood cell. In sickle cell anemia, that that mutation, that valine change, causes the hemoglobin molecules under stress to polymerize. That is, they make a long chain. They attach to each other, and they make a long chain. And as they make a long chain, we already know that red blood cells are flexible, right? They can blow up and crenate, and they can swell. So we already know they're flexible, and those hemoglobin molecules now make a long polymer, a long chain, which causes the cell to change its shape, blocks the blood flow, and these individuals have episodes where damage is being done to tissues. If the blood flow is blocked to the brain, to the lungs, to the kidney, it could be significant damage to the tissues and even death. If the blood flow is not going well to the finger, they might have painful episodes, but they will survive that. But each and every time these blockages occur, they're sustaining more and more damage. And over time, their internal organs become a mess. So those individuals have to reduce their stress levels because stress causes the sickling. They can't go to high altitudes. Higher altitude also causes the sickling. They can't fly without potentially causing a sickling event because that higher altitude in the plane can trigger this. And when they are having an episode, they have to get transfusions where they try to basically get rid of all the sickling blood and get in fresh blood. It's a very harrowing disease. The point of all this is all about what? One nucleotide that went wrong from a mutation. These individuals are born with this. They inherited this. So for whatever reason, right, these individuals carry this. Mom is fine. Mom's a carrier. Dad's a carrier. Life is good. But their kids, some of them, can have a full-blown sickle event, and that can really change their life. I've already talked about this. This is just a review. Another one of these ideas where there's a very quick mutation, or a mutation which causes a very quick stop. I already showed this slide to you before. I've got it duplicated. So that's the end of your chapter 3. We've still got a lot to go, but that was like a double chapter. There's a lot of information there. It was like 2 PowerPoints. The first PowerPoint was on cells and organelles. The second half of it was on transcription, translation, a little bit of replication, the tumor story, the cell cycle, the mitosis. And the bottom line is that changes in the DNA can change proteins. Proteins, when they're changed, can cause disease. That's really the bottom line. Quick review on the exam next week. You better expect to be able to label the organelles. So don't look on your paper because I think the answers are already there for you. But just look up here on the screen as I go through these and tell me number 1 is what? The nucleolus. And its job is to make ribosomal RNA. It's the building block for ribosomes, absolutely. Number 2 is the nucleus. There is where we find the DNA and the chromosomes. Number 3, the arrows pointing to the little tiny studs. So those little tiny guys on there are the ribosomes. They're all about making proteins. Number 4, I have no idea. We'll keep going. I can't label it in any meaningful way. I don't know if it's a lysosome or a peroxisome or what it is. It's just a little vesicle. I wouldn't have you label that. Number 5, now we're looking at the membranes. It has ribosomes on it, so that is the rough ER. Again, you're thinking about the production of protein. Number 6, Golgi, all about what? Not the production of proteins, but the packaging, the rearrangement or the packaging of the made proteins. Number 7, yeah, some part of the cytoskeleton. It could be a microtubule or microfilament. Number 8, smooth ER. It is ER, but without ribosomes. It's not making proteins, but it is instead making lipids are made here. Okay, lipids are made on the SER. Number 9, good old mitochondria for the production of ATP. 13, centriole. Each little part is a centriole. What are those important for? Yeah, from those centrioles, the mitotic spindles are formed, and they're critical for cell division, critical for mitosis. Number 10, again, it's a vacuole. I don't know if it's a lysosome or not, okay? I can't tell a lysosome from a peroxisome. You know the difference, though. Lysosomes are involved with what process? breaking down things and apoptosis, and peroxisomes are in the job of detoxifying and breaking things down by oxidation. So that brings us to the end of that chapter. I know that my numbers are off on the chapters. Again, that was 3A and 3B, if you will, and that makes up a good half of this exam. Okay? So those two PowerPoints make up about a half of this. And then we move into, again, my numbers are off here. Make that chapter 4 for you, and I'll get this updated. So this is chapter 4 for your new text for the Martini text. Questions on anything related to the cell organelles, cell cycle, transcription, translation, any of that conversation? If you've taken a general biology course, you've heard this story in different versions, different levels of depth. If it's been a while since you've had a general biology course, you may not be that familiar with this story. So some of you are more relaxed than others when it comes to transcription and translation and tRNAs and that conversation. Anything for me, though, right now, question? Let's take a quick break, about 5 minutes or so, and then I'll come back and we'll go into this next chapter. Okay, so let's get moving. This next chapter is actually chapter 4 in your Martini book. And this chapter is all about histology, tissues, not nose tissues, but the study of tissues. And let's start off with sort of an overviewing idea that I want you to appreciate. When you get a chance, and I know when you're in a class, you're like overwhelmed by the details, but at some point in the future, hopefully you'll look back and think about some of these things and say, wow, this is really cool stuff, right? How is it that an egg and a sperm can come together to make the zygote, and from that fertilized egg or from that zygote, we are able to form everything about us, right? Trillions of cells in the right place, doing the right job. And then how is it that as an adult, we're then able to create a sex cell, a gamete, which then can unite and give rise to yet another individual. So the overall story here is pretty cool. So we'll start off with the zygote. Again, the zygote is just the union of the egg or the sperm. Other books just call it the fertilized egg. And in lab, I mentioned to you that the cells are dividing very, very quickly. When we get to a point called a blastula, when we were looking at cells undergoing mitosis a couple of weeks ago in lab, we were looking at fish blastula, rapidly growing balls of cells from a fish embryo. And the next step in that is the gastrula. Now, the blastula is like a ball of cells that begins to hollow out, and then the gastrula is like, take a balloon, imagine a balloon, and you stick your fist up into the balloon. And what was on the outside now is drawn up inside the balloon as you push your fist up in. And that's sort of what the gastrula is. It's where we go from being a round ball to start to create more of a three-dimensional being. And in that gastrulation, in this process, 3 different layers of cells are set up. Okay, these 3 different layers. Now, these are called the germ layers. So we have, first of all, the ectoderm. It's in blue. Ecto, we know, means toward the outside. So this is the outer layer. The outer layer of that gastrula, of that blastula, will become your epidermis. That makes sense, the outer layer of you. It'll also become your nervous system. And that may not seem as obvious, but your brain and your spinal cord will also be derived from this ectoderm. The middle layer is the mesoderm. Meso, middle. So from the middle layer, you're going to get your muscle as well as your connective tissues. And you know that includes things like your bone and your blood. There's a lot more to this. This is a very simplified approach to this. And then the endoderm, the inside layer, will become the lining of your cavities. So it'll become your digestive tube, what will become your digestive system. It'll become your pharynx and your respiratory tube. All those tube-like structures are derived from the endoderm, the within layer. In addition, there will be a few cells off to the side that that will eventually become or create your germ cells. So there'll be cells that are going to eventually, in the male, produce sperm, and in the female, produce eggs. This is just a blow up of the same thing of those three layers. So I definitely want you to appreciate the three layers, ecto, meso, and endoderm, and know the basic organs that are derived from each. So again, the ectoderm, the outer layer, creating the epidermis and your central nervous system, your brain and spinal cord. The endoderm, the inside, that's easy, think the tubes. So the tube going through is your gut, and the tubes going into your lungs, your respiratory system. And then finally, the middle layer, making up sort of everything else, muscle, as well as your connective tissues. Now, it seems like, I haven't heard any legislation coming down the pike this time, but it's oftentimes that as voters we're asked to, or will be asked to vote on stem cell legislation. And it's really important that as a population, as a voting public, that we understand what stem cells are and what they are not as we're in this conversation. So we see here this continuum, and sperm and egg come together, and we make the fertilized egg. The other word for that would be the zygote, right? And now we get to a morula stage. Now, the morula is an intermediate stage. It's like a solid ball of cells. This is derived from a word that means blackberry, basically, like a little mulberry or a little blackberry. So the cells are like this little solid little ball. And then we saw that in the blastula stage, there's a hollowing out. And what's interesting here is that the cells that are here, right, the cells that are out here, they will actually become the embryo itself. The cells that are out here on the periphery will go on to help form the placenta. So this is about that differentiation story. Already, at this early point in development, some cells have already been given their assignment. Some cells will go on to become the placenta, which won't become part of the body itself. And some of the cells have already been chosen and differentiated to become the embryo itself. And from those cells of the embryo, all the other organs and tissues of your body will be developed. So already we see here that some, quote, differentiation has occurred. But we take these cells from the inner cell mass. So these cells in here, right, will eventually become your brain, your kidney, your liver, your every other kind of cell in your body. And we can put them into a petri dish and grow them. The hope is that we'll someday be able to take these cells and create from them any kind of cell that we need. So we would learn the tricks to make these cells into blood cells. We would learn the tricks to make them into cells of your stomach or of your nervous system or of your pancreas or of your liver or of your heart muscle. We're not there yet. But that's the hope, that we can take these cells and trick them chemically, biologically, to create whatever we want them to create, and then use those cells to insert them back into a patient as a form of therapy. Right? So that's what the hope of stem cells is all about. Now, we're not there yet, but that's the idea. What's the controversy here, though? What's the controversy? Well, to get these cells, right, what has to be destroyed? An embryo. There's no doubt it's an embryo. I mean, you can argue ethically what those cells represent, but if you look at the life cycle of a frog, right, it tells you the life cycle of the frog starts when the egg and the sperm from the frog meet to form the embryo, to make a tadpole, to make a frog. So no different in you and me. Our life cycle, the human life cycle, definitely starts at the formation of the zygote, right? And we move around through the life cycle. So there's no doubt that this is an embryo and that it has the potential of life. And the only place we've had, up until a few years ago, to get these cells for research was from here. And it created the ethical dilemma of destroying life. Okay? So how have we gotten around this? Well, regardless of what you hear, to date, these embryological stem cells have done nothing to cure disease. We've not yet gotten to a point where we can do something with them in a meaningful way. But there was a moratorium a while back, and no more stem cells were allowed to be created. The ones that were used were already in place for the ones that were used. And during that time of sort of drought, we learned that we can now find adult stem cells. Now, you also hear about adult stem cells in cord blood. When babies are born now, you have the option of taking some of that cord blood and storing it away. We don't know what we're doing with it yet, but it's going to be stored away for a day when maybe in the future we know what to do with those cells. And we can now take adult stem cells and do all the same tricks, or at least potentially do all the same tricks. So we have been successful with using adult stem cells for some diseases like fixing a damaged heart, creating some cardiac muscle cells. And so this is probably where the wave of the future is. So I suspect that in the next 10 years, this whole idea of having to destroy embryos to get stem cells will be a moot point. That we will have figured out how to get stem cells from ourselves. You can even take stem cells from your skin. Right now, I could take stem cells from you and me, go down into the layers of my skin, and I don't know which cells down there are stem cells, but some of those cells in my skin are there to repair damage. So if I cut my arm, cut my hand, those cells can regenerate and make new skin. If I take one of those stem cells from my skin, about 5 years ago, a group figured out how to deprogram skin back to an earlier embryological state and then forward it to muscle. So we used to think that a skin cell, once a skin cell is always a skin cell, can't do anything else with it. We've now been able to take skin, roll it back, and then roll it back forward again and make muscle from it. So we are maybe closer to creating cells from adult stem cells for therapeutic uses. Now, why do you think we can't yet make organs? If you look back in this picture, everything on this picture is talking about taking cells, right? These are all cells down here, and using the cells. You don't see a liver or a kidney or a brain here. You see cells, cells, cells. Why do you think, at least so far, we're not able to take a liver cell and have it create a liver? Why can't I take a cardiac muscle cell and make a heart? Vascular system's part of it, but there's something even more fundamental going on. Yeah? When does a heart form? In the body, in relationship to all the other organs. So as the heart is forming, it's right next to the lungs and to the stomach and the liver and the brain. And certain genes are turned on early in development in the making of that heart. So we would have to figure out what are the signals, what are the hormonal signals, what are the chemical signals being received by those heart cells to make the heart not only in what we call sequence of space, right, spatial relationship, but also temporal relationship. We would have to know exactly what timing was necessary and the sequence and the spatial relationships of all the other organs in order for us to create an organ. We're not there. Now, we have gotten pretty good. We can make skin, right, but it's flat. It's kind of boring. Skin's not as complex as any other organ. So we have kind of been able to make some skin, and there's hope that we can make some, quote, fake blood, sort of a synthetic blood. But other than that, right, we're nowhere near making organs from cells. And hopefully we'll never have to ask this question, right? As we start doing genetic engineering, we start pushing genes from one organism to another. Hopefully we never have to ask, well, you know, is this dude human? Because he kind of looks kind of creepy, doesn't he? Yeah. You got a friend, you have anybody who you know that looks like that? Right? I have a friend who looks like, just, wow. It's almost like his face. It's really weird. Okay. Okay, everything but the nose. Okay, now, where are we? Again, my chapter numbers here are off a little bit. Organelles and transcription, translation. This was all in Martini. This is all chapter 3. So these are both chapter 3. And here we are now in what is chapter 4 for you, okay, in the Martini book. you know. Okay? So a lot of what I'm about to say, you've already been introduced to in lab. So I want you to kick back, kind of say, okay, I know that, I know that. Oh, that's a new thing. I better focus on that. So let's just kick back and learn a few things. Now we know that tissues are formed from cells that come together and they carry out some common function. And you know there are 4 types of tissues: epithelial, connective, muscle, and nervous, in no particular order. And this is new for you. Epithelial tissues are formed from all 3 germ layers. That is, some epithelial tissues are formed from the ectoderm, from the mesoderm, and some from the endoderm. And that kind of makes sense when you think about where does one find epithelial tissues? Covering the body, ectoderm, lining your organs in the middle, mesoderm, and lining your gut tube, endoderm. So it makes sense that you would have epithelial layers derived from all 3 of those germ layers. Connective and muscle, all from mesoderm. Right? Connective and muscle tissue, all from mesoderm, and nervous tissue, all comes from the ectoderm, from the outer layer. So let's start off with epithelial tissues. We spent some time in lab talking about these. We know that they're lining, coating, covering us. Your glands are also largely epithelial. We know that these tissues are composed of cells that are tightly packed together with very little, if any, space in between them. That is, there's no or little extracellular matrix, no space in between the cells. Very few, if ever, are there blood vessels penetrating through the epithelium, and that's a good thing. As I mentioned in lab, you would not want to scratch your skin and bleed. You would not want to eat something and bleed down your esophagus, so those epithelial layers are blood-free.
When you have something like a varices or something?
That's the exception. Okay? So we'll get to that in a moment. Now, we know that epithelial tissues are bound together tightly. And I've also introduced the idea of apical and basal, so that cells have a free surface and cells have a basement layer, or they have a polarity. They know their north pole from their south pole. We also see that during mitosis. We talk about how the chromosomes lined up on the Mediterranean, on the metaphase equator, right? So we know that cells have a sense of direction. They know their top from their bottom. These cells are also connected tightly to each other. So there must be something that is grabbing and allowing these cells to reach out and hold on to each other. We know this if we look at epithelium. 2 ways of describing epithelia. One has to do with their layers, right? 1 layer, simple epithelium. 2 or more layers, stratified. That's one word we'll always use in describing an epithelium. The other word we'll always use is the shape of the cell. 3 basic shapes: squamous, cuboidal, and columnar. We've looked at 6 different epithelial tissues in lab, and those we'll get to know and love even better. At the bottom of the epithelial layer, there's going to be a layer called the basement membrane. That basement membrane is what the epithelial layer is sitting on. And then what do we know is below that? Below the basement membrane, one would find what? Connective tissue, right? So the epithelial layer always sits on top of connective tissue. The basement membrane becomes the divider between the 2 tissue types. There we are. Well, somehow I can't find my cursor. Okay. Okay. So I've also told you that the epithelial cells are attached down to the basement membrane, and there are no blood vessels. So the other way of saying that is epithelial tissues are avascular. blood is how cells get their oxygen. Blood is how cells get their nutrients. So how must epithelial tissues get their nutrients then? It has to come from neighboring tissues, from neighboring blood vessels, and we love that process called diffusion. So we talked about diffusion, that is molecules moving from an area of higher to lower concentration. So the reason we suffered through those labs early on in the course is so we had a better understanding of the highly innervated, that is, it has many nerve endings going to it, so that you are aware of temperature, touch, pain. Those things are all largely coming from your skin. Now, there's even more nervous tissue deeper. So in your skin, remember the outer layer is your epidermis, and that is epithelial, and then the dermis is made of connective tissue. So yes, you have some nerve endings in the epithelial layers, but you have even more nervous tissue down in the dermis, in the connective tissues. Now, these cells are constantly being replaced. So they're highly mitotic. They have a high regeneration capacity. They're highly mitotic. They can be damaged and replace themselves. You can burn your tongue, scrape the inside of your cheek, cut your knee, and you know that your skin and your epithelial layers are going to quickly regenerate. They create physical protection, especially if you think of your epidermis, and your epidermis is creating a stratified layer on the outside. But don't forget that epithelial tissues are also creating a barrier. So if you think about your serous membranes, your visceral and parietal layers, or if you think about the lining of your oral cavity or the lining of your nasal passages, yes, they are also separating and also allow for some selective permeability. So what is allowed into or out of the digestive system is largely defined by the cells lining the digestive gut. Now, epithelial tissues are also the major component of your glands. So we need to talk about glands here a little bit as well. Now, glands come in two basic types, endocrine and exocrine. The word tells us what they do, and we already know what crine means. So crine means what? To secrete. So endocrine glands must do what? Secrete within, and you know that the endocrine system are all glands that secrete hormones. They're within the body. Whereas exocrine glands are going to secrete out of the body. And this is going to be things like your sweat glands, your tear glands, even your digestive juices, even though they occur inside your gut, have a direct connection to the outside world, and so they're also considered exocrine. Again, those nerve endings in your epithelia is helping you, know your environment, hot, cold, painful type things. And you have a constant supply of information coming in through those nerve endings. Let's take a look at a simplistic cartoon of the epithelium again. We know that the epithelium is sitting on the basement membrane. There is, however, something extra I want to teach you here. So here's our basement membrane. But the basement membrane is actually 2 layers. We can divide it up even more. Now, in anatomy, anatomists get around and they decide, okay, let's break it up and call it something else. So the basement membrane is that layer, but it can be further divided up into the basal lamina and the reticular lamina. Now, you know a little bit about that word lamina. What does it sound like to you? What other word have we talked about that sounds a little bit like lamina? Lamella. And the lamella were the layers within the osteon, the layers within the bone structure. So the lamina is a layer, okay, and it's called the basal lamina. So it's the layer in the bottom of the epithelial cells. And then below that is the reticular lamina, and we know the word reticular means a network. So basically, the basement membrane is a double layer, half made by the epithelial tissues above. So the epithelial cells are making the basal lamina. The connective tissues below are making the reticular lamina. They're coming together, they're kind of gluing together to make that thing called the basement membrane. So it creates a barrier between these layers of tissues. We already know this story, so I'll pass forward on this. You know about, again, in this picture, the basement membrane just shows as a single layer, right? But keep in mind it is a double layer, basal lamina, and below it the reticular lamina. Now you also know that these cells are tightly packed. That's one of the basic ideas of epithelial tissues, tightly packed cells. So they must be held together by something. The things that hold the cells together are collectively called the cell junctions, or the intercellular junctions, things that hold between the cells. 4 types of these cell junctions. The first, tight junctions. Now, tight junctions are described as circling up near the apical surface. So this is the top of the cell. The apical surface of the cell. Kind of looks like Charmin, right? Looks like 2-ply toilet paper and the little stitching, okay, between the plies. So the layer that comes around and knits around toward the apical surface is called a tight junction. Now, what these tight junctions are doing is they're holding the cells together, but they're also making sure that molecules can't go between the cells. Right? So if a molecule is going to enter into your digestive system, it must be welcomed into the cell, and it can't squiggle around the outside between the cells. Then there are the adhering junctions. They also form a circle around the cells, but they're a little bit deeper. So down in here would be the adhering junctions. Thirdly, there are what's called the desmosomes. These are like little snaps or little buttons, or I think of it as my rivets on my jeans. Where do you find rivets on your jeans? Areas of higher stress. That is at the pockets, right? So little extra adhering, little spot welds. And that's what these are. One half of the button is provided by one cell. The other half of the button is provided by the other side. And together they snap together. And it creates, again, an area of greater resistance to stress. And then finally, gap junctions. Gap junctions are formed, they're like little tunnels, little channels between the cells. And this is the way by which molecules can communicate from one cell to the next. So gap junctions are made with these little proteins called connexins, and they allow for glucose and amino acids and nutrients to travel from one cell to the neighboring cell to the neighboring cell. So it's a way of communicating between the cells. But it also creates, again, another way of connecting the cells together. So all 4 of these, I think, are better represented on this slide. So put a little note to yourself. This is the slide that I'd rather have you look at because it has all 4 of these on one view. And collectively, they're called the intercellular junctions. And again, up here, we have the tight junctions, the adhering junctions down below, the desmosome buttons, and the gap junctions, all shown on one picture. I also like this picture in that it shows both layers of the basement membrane. So it does show the basal lamina and the reticular lamina separately in this picture. Okay, so we know cells are tightly held together. We know that there are simple epithelia, only one cell thick. This tells us that this epithelium is not going to be very protective as far as stress, but it's certainly going to be good for getting very small molecules across a very small area. So simple squamous, for example, would be where? Where would we find simple squamous? In the alveoli, in the lung. Simple columnar was where? Simple columnar was in the intestines, and simple cuboidal was in the kidneys. But all of those simple epithelia have what? They all are involved with some sort of filtration or absorption or secretion function. They're not very protective. They're only one layer thick. So they're going to be found in places where there's minimal stress on the tissues. Versus, as you know, stratified. And this should not be one or more, but two or more. Two or more layers, typo. So 2 plus layers. Now, in a stratified epithelium, the deepest cells, the basal layer, are the only ones attached to the basement membrane. So down here, only the basal cells are attached to the basement membrane. The rest of them are attaching to each other, right, and are moving toward the surface. So this represents a good view of your skin. But also, we find stratified epithelium where there's any kind of mechanical stress or abrasion. So this will include the esophagus, the anus, and the vagina. There's that one other type, the pseudo-stratified. It is its own little classification. It looks at first glance to be stratified, but if we look again, or we could tease it apart, we would note that every cell does go down to the basement membrane. So it's really just one layer of cells that are attached to the bottom, although they look to be stratified. And where did we find that again? Where was pseudostratified? You should be thinking pseudostratified is the trachea and the respiratory tracts. And then we have shape. And again, we know this story. We know that they come in 3 basic shapes. There's one, though, that I did not tell you about, and that is transitional. I'm not going to have you recognize it, but I do want you to know about transitional. This is an epithelium found in the bladder. It's found lining your bladder, and it, quote, transitions from a very thin layer to a more stratified layer. So as the bladder fills and voids and fills and voids, it transitions with that stretch. Okay.
What would the shape of the pseudostratified be? Is it all different?
Pseudostratified always goes with the word columnar. So pseudostratified, columnar, those words. Now the cells do have different shapes, but the majority of them are columnar. So those 2 words always go together, pseudostratified, columnar. So let's go through a real quick view of these. Now, the images here are different than what you have in your book. I think it's really helpful to not look at one image and memorize the image as much as to learn how to recognize these things. So that's why I really want you looking in your Martini book, looking online, looking at your Amerman lab manual, and get your eye to see this. Now, on this, you're like, I don't know where he wants me to look. I'm going to always point, but in this particular image, what I want you to look at is just this layer up here at the very, very top. And that layer is definitely one layer of squamous cells. So that is simple squamous. Now, in the lab, I told you that that is found where? Simple squamous was for gas exchange in the alveoli. But I also want you to realize that this simple squamous is what makes up your mesothelium, the alveoli, but your pleura, your pericardium, your peritoneum. Those are just simple squamous layers. They're extremely thin layers. They're on the outside of your organs and lining your spaces. Also, the inside of your blood vessels, a special layer called the endothelium, and that's also for gas exchange. So in your capillaries, you have an endothelial layer. Again, a simple squamous layer. When you hear a simple squamous, think gas exchange. Simple cuboidal. When I say simple cuboidal, what comes to your mind? It needs to be the kidney. And think about the tubules within the kidney. And in this view, in the lab, I showed you a circular arrangement. Here we're looking at a longitudinal section of a tube. And you can see these beautiful cuboidal shaped cells. So this is the lumen here. And you're looking at the tube cut open longitudinally rather than looking at it transversely like we did in lab. Simple columnar comes in 2 different types. We only looked at one type in the lab. We looked at the second one. We looked at non-ciliated. I never mentioned it. But I did tell you that the simple columnar that we looked at in lab had microvilli associated with it. So the microvilli, again, were there to increase the surface area, and we saw this type of tissue in the small intestine. So small intestine, microvilli. Now there are some simple columnar that have cilia associated with them, and those we'll find in the uterus and in the fallopian tubes, the cilia help to coax the egg down the fallopian tube. Also, you'll find this type of ciliated epithelium in the bronchioles, in the air passages. Again, that cilia trying to keep particles from getting down into the tissues. Keep knocking myself out of here. The next one, this is the simple columnar. Now, do you see on this that right along here there's a fuzzy edge? You really can't see anything distinctive, but that fuzzy edge is microvilli. You cannot see individual microvilli. This would be your lumen right here going through the intestine. Again, those microvilli are there to increase surface area. And you can also see beautiful examples in this tissue of what? What are these cells? Yeah, those goblet cells making the mucus. Very obvious in this particular sample. So we would call this non-ciliated because it has microvilli. I think I mentioned at the beginning of chapter 3 that no one cell is going to have both cilia and microvilli on its surface. So it's either one or the other or neither. Microvilli are there to increase surface area. Cilia are there to push things. They have a different job. So here, in this example, can you see these little hair-like extensions? Right? So these are cilia. I can see individual cilia. And this would be, as the picture tells us, in the fallopian tube. And those cilia are there to assist the sperm in their movement and also to help the fertilized egg and the embryo to start going down toward the uterus. Stratified squamous, we looked at a couple types in lab. We looked at non-keratinized versus keratinized. The first one here is non-keratinized. What do you see? Your eye has been trained to see something. What are you looking for? What's at the surface? I still see nuclei. These cells are still alive. So this is a stratified epithelium. Here's your basement membrane down here. Below it would be your connective tissue, loosely packed cells. Above it, tightly packed cells, alive and well all the way to the edge. They did not undergo apoptosis. And we find these in mucous membranes. A mucous membrane is any part of your body that has an opening to the outside. So this would be the vagina, the oral cavity, the anus, down the throat a little bit. All that would be this non-keratinized stratified squamous. Versus this tissue. Now the staining is a little different than what we saw in our lab. The blue and the red, don't be thrown off by that. But what you see here is the blue down here is the border of the connective tissue. So the connective tissue is in blue. There are layers of cells. Down in this layer, they are alive. They're fine. They're living, they're dividing, they're moving. About halfway up, you see a dramatic change in their look. And about here, they begin to undergo apoptosis. And from that point up, they are dead cells. Those little spaces you're seeing are basically gaps. A lack of nuclei. You see no dark nuclei at the surface. And these cells filled up with a protein called keratin, which creates a very strong waterproofing type outer layer in our skin. So we did see those two types of stratified epithelium. I should go back and remind you that the key is this. When looking at a stratified epithelium, you always look at the shape of the cells at the apical surface. So this is a stratified squamous because at the apical surface those cells are flat. Now there's a couple more examples that I'm going to show you that you do not need to recognize. So don't spend as much time with these, but I did want you to know that they are in the body. In the lab we cross these out. So you're not going to be asked to recognize them, but I want you to at least appreciate that they exist.
The keratinized, where did you say those were mostly found?
Keratinized is only your skin.
Only in.
Yep, the outer layer of your skin, your epidermis, is stratified squamous keratinized. So there is stratified cuboidal in your body. We're not going to look at it. I'm not going to expect you to recognize it. But you can look at this on the bottom and appreciate that here's your basement membrane, connective tissue and muscle down here, and you see at least 2 layers of nice cuboidal cells. Okay? So that is stratified cuboidal. It's found in some of your sweat glands. There is also stratified columnar. Okay, 2 layers of tall columnar-like cells, and they are found also in some of your exocrine glands. Pseudostratified columnar, this one's on our list, so this one is important for you to focus on. This pseudostratified columnar is in the respiratory tract, as it shows us here in this picture. And what we see here, there's my connective tissue border, my basement membrane, connective tissue down below. And I see beautiful examples of goblet cells. And I can see individual cilia. So I'm looking in the trachea, in the respiratory tracts.
Is there a reason that goblet cells, well, at least they don't appear to have nucleus? Or are they just different?
They don't appear to have, but they certainly do have a nucleus. But in our pictures, the mucus is much like the adipose. The mucus kind of gets washed away, and what we're seeing is just the hollow evidence of a cell. So the goblet cells, we're just seeing the evidence that it was there. I mean, it's there, but it's largely nondescript. It's more these big white billowing spaces between the cells.
So if you were to try and look at a goblet cell under a microscope, just like a singular cell, you would see basically like adipose, like fat.
It's different, but there would definitely be a nucleus. Okay, every cell has a nucleus. It just is not always as obvious as other times. And then that transitional epithelium, I told you about it. Again, you don't have to recognize it, but this, and you can maybe see why I don't have you recognize it. What might you get confused with here?
Highland cartilage.
Maybe Highland cartilage, but even maybe the pseudostratified, a little bit, just a little bit. So I don't ask you to know this one, but this one changes its identity as the bladder fills and as it empties. Okay, so we know that we have different types of epithelial tissues, and glands are also largely epithelial. So let me describe glands to you for a moment. Endocrine, we know, does what? Endocrine glands secrete hormones within. Endocrine, one of their characteristics is that they secrete their products directly into the bloodstream or into the body. So we see that there are no ducts. They lack ducts. They lack tubes because they don't have to release these products to the surface of the body. And they go straight into the bloodstream or straight into the fluid around the cells. So this is how our hormones are released. Versus exocrine. Exocrine glands have to get their products out of the body. And in order for them to exit, there's going to be a duct, a tube, that's going to carry that product to an epithelial surface. So this could be a sweat gland releasing sweat to the surface of your skin. This could be a salivary gland secreting spit into your oral cavity. Let's take a look at a cartoon representation of a gland. Whenever I see this picture, I imagine I'm flying over a city and I'm in sort of a neighborhood where there's lots of little cul-de-sacs. That's kind of what I see here. I see, a major road coming into the neighborhood, but then all these little side streets and these little cul-de-sacs, and all that's really missing are little, swimming pools, right, sitting outside. So let's zoom in to one of these cul-de-sacs. And what I see now is this. The cells here are epithelial cells. They're releasing their products. They're releasing their products through exocytosis, right? So these cells are making their products. They're being released by exocytosis, and they're collecting in the lumen, and they're traveling out of the body to the epithelium surface. The cells here on the edge, all these cells that are producing the secretions are part of what's called the acinus. The acinus. Now let's describe these exocrine glands. They come in different forms, different types of secretions, different methods of release. So let me go through these terms with you. First, I'll start with morphology. Morphology is the shape of the glands. I'm not going to ask you to recognize these or to name these, but I think you can appreciate that glands, some of them are very simple to extremely complex. Just know that glands come in many different shapes, but the details of which we're not going to worry about. What I do want you to worry about, though, are the different methods of secretion. The first one I'm going to describe is merocrine. They all have crine in them. They're all secreting. And in merocrine, we look at the cells here, and we see the cells down here in the acinus, and they're releasing their products, and the products are going up to the surface. And this is basically how your salivary glands work. So you've got these salivary glands in your oral cavity, and they're releasing spit right into your oral cavity. And that's why your dental hygienist has to work harder. In fact, where do you have more plaque buildup, more calculus buildup in your mouth? Do you usually have a tougher spot in the back of your lower teeth? Right? A little bit more buildup. The reason is there's salivary glands that dump their products right back there into your mouth. And so those protein products kind of accumulate and create layers that are hard to get off. There's also other ones back between some of your molars. So again, that's another area that's more difficult to clean and for the hygienist to especially pay attention to. So the merocrine glands, again, we just see normal release of the product. This is, again, your saliva. This is your tears. This is your gut juices, right? Your stomach is releasing things via merocrine. Then we have holocrine. Now, this is a little bit different. Here are the acinar cells, but notice that when these cells release their products, they're not just releasing their products. The cells are actually breaking off and are disintegrating. And that makes this stuff that they produce very sticky. This is the way that your oily glands on your face secrete their products. Is it any wonder our pores get clogged up? Because they're not releasing a nice liquidy stuff, they're releasing oil plus gunky sections of the cells. The way I remember this is that in holocrine, the whole cell, right? The whole cell is destroyed in the process. And so these secretions include ruptured dead cells that tend to clog up our pores. Now back up then, the way I remember this is that if this is holocrine, back here, these cells stay, this is goofy I know, but these cells stay merry. They're not dying, they're just releasing their product into the duct, and they're not being destroyed in the process. So merocrine, right, those cells stay merry, they're alive, versus holocrine, where the whole cell dies as part of the process. The third type is apocrine, and we know the apical surface is the free surface. And in apocrine glands, what happens is that here's our acinus, and just the apical portion, right, just a portion of the cell, the apical side pinches off and is released into the lumen. This is kind of intermediate. This is how breast milk is produced. It's intermediate between merocrine and holocrine. Just the apical portion pinches off and is released along or as part of the milk. Okay, so this is your milk secretions as an example. And then finally, your secretions can be very serous, which is a fancy word of saying very watery. They can be mucousy, thick, or they can be what's called mixed. So serous secretions are very watery. If I say your serous membranes, what comes to mind? Remember, the serous membranes were around your heart and lungs, and they allowed for movement of your organs to slip and slide over each other. So they have a very watery-like secretion involved. Mucus glands would be very thick, and your salivary glands are considered mixed. And we've all experienced this, right? When you're hungry, a lot more watery secretions to your mouth, and you're anticipating the meal for digestion. And when you're scared, all of a sudden your mouth gets really dry and sticky, and you're secreting more mucousy kind of stuff. So we know that the salivary glands can be both watery, serous, and mucousy in their secretion. That's it for epithelial. I spent the most time there. Let me also run through a quick reminder about connectives. Again, this is what we know the best because we've had this in lab. Any questions on epithelial, especially about glandular secretion, since that is new?
Is there something that the cell knows the difference with the mixed? Why does it change and what causes the change?
Yeah, different cells, and we'll get into the sympathetic and the parasympathetic nervous system. We'll get into rest and digest and the fight or flight responses later on. And actually, that's part of it. So under different signals from the body, when you're about to eat, when your body's anticipating digestion, the secretions are more watery. When you're scared, they're more thick. Okay. We'll talk a little bit more about those signals later on. But not today. It'll be in the nervous system. Anything else related to glands or epithelial? Okay, so connective tissues, we know the story. We know that this is a very diverse group of tissues, that they're all somehow holding us, binding us, connecting us in some way. And it includes a very broad group like the dermis of the skin, ligaments, tendons, blood, cartilage, bone, fat. We know there are a lot of members to this. Now, connective tissues do all sorts of jobs. They are protectors, right? Bones protect our organs. Fat protects our organs. Cartilage helps to support us as well. Ligaments hold us together, our bones to bones. Storage, our fat is being stored by adipose. Our calcium is being stored by bones. Transportation, blood is transporting all sorts of things. And we don't want to forget about immune functions. Some of your white blood cells are helping you fight off infection. So lots and lots of different types of and different functions for the connective tissues. This table I like. It kind of breaks up what we know and adds a couple more details to it. So we can say that all connective tissues have a common origin. I don't like this word, though. Here it says mesenchyme. That's a fine word, but I haven't used that word. The word I used was mesoderm. Right? So just know that all of your connective tissues are derived from that middle layer, the mesoderm, rather than that mesenchyme word. And your connective tissues come in three basic types. Connective tissue proper. Kind of a weird name. But then there's the supporting connective tissues and the fluid connective tissues, or tissue. So let's start there, because we know this one. There's only one fluid connective tissue, and that is blood. Got that, check. There are two structural connective tissues, or supporting connective tissues, and we already know those as well, cartilage and bone. Under bone, you guys will see next Monday in lab that there's compact bone and spongy bone. The only one we're looking at under the microscope is compact. So when I described to you osteons and canaliculi and the lamellae and all of those structures that we saw in bone, that was compact bone. Then under cartilage, there are three different kinds of cartilage. We only looked at hyaline, but there's also a fibrocartilage and a elastic cartilage that we'll deal with later in the course. But for now, just recognize hyaline cartilage. Then the other side, there are connective tissues that are considered loose and some that are considered dense. Under these, the ones that you need to focus on, we saw dense regular, didn't we? Dense regular was the type of connective tissue that makes up your tendons and ligaments. The other two, we didn't look at, so you're not going to worry about them. And then over under loose connective tissue, You saw that areolar. Remember, that was that confetti-looking loose connective tissue. And adipose is also considered a loose connective tissue. The reticular, again, don't worry about reticular. So again, we're looking at the same 6 that we already know. That's what you want to focus on. You already know what drives this group. You know that all of the connective tissues, blood, bone, fat, and cartilage, are united by the following characteristics. They all are composed of loosely packed cells. They all have around them a ground substance, that is the non-living stuff around the cells, and that within that ground substance there are protein fibers. Those protein fibers come in 3 types: collagen, elastin, and reticular. Collectively, the ground substance and the protein fibers are called the extracellular matrix, or the ECM. Remember, connective tissues have a lot of space between them, right? The cells are loosely packed, so the stuff between the cells is collectively called the ECM, the extracellular matrix. Now, tell you what, the next 2 slides I'm not going to even worry about. So this idea of wandering versus fixed, just when you look at I don't care about the idea of wandering or fixed, but you do recognize some of these names. If I say to you adipocyte, you know that's the cell that makes up fat. If I say to you fibroblasts, where do we see fibroblasts? We saw fibroblasts in the loose and in the dense, regular connective tissue. And we won't worry about the wandering cells for now. Now, I introduced these different protein fibers. In lab, there was the collagen fibers. And I told you, whenever you think of collagen fibers, think strength. So this is the steel of your tissues. Very, very strong. Your tendons, your ligaments, and your bones have large amounts of collagen. Elastin, a more stretchy protein that allows your tissues to change shape a little bit, like your nose and ear. And reticular fibers, which are more of a mesh-like or scaffolding arrangement. So again, under loose connective tissues, the only 2 you need to know, areolar and adipose. Loosely packed, right? There's just not a lot of stuff here. It's more of a very diffuse, when we say loose connective tissue, we're talking about a looser arrangement of cells, a looser arrangement of the fibers. You know and love this one, right? So this one's your areolar connective tissue. The other word I had for this in lab was just loose connective tissue. And what did you see? Party, confetti, right? And at the party, who was there? Loose people. It's just a way to remember it, right? So I see confetti at the party, loose people. There we go. It'll remind you of this. What kind of cells do we have here? Fibroblasts. And what are those fibroblasts making? Fibers, right, of different types. Then adipose. You recognize probably the bottom picture better, the bottom inset, as being what you saw in lab for adipose. Again, this is a loose connective tissue, and what we're left with under the microscope are spaces. The fat has been dissolved away, and we just see spaces. The adipocytes are basically empty. If you could see it, though, in real life, if you can imagine, it would look more like this, right? Little globules of fat within those cells. There is reticular connective tissue. It's not one of the ones we need to worry about. But what do you see here? What does it look like to you?
Blue cheese. I don't know.
Blue cheese, or I kind of see broken chicken wire, right? And the idea is this is reticular. It makes a scaffolding or a mesh-like. I like your blue cheese idea, though, right? It kind of looks like that. But sort of this chicken wire kind of idea. Now, under the dense connective tissues, we've already seen this one. This is dense regular. And what we're seeing is a densely arranged layers of what? What are all these little swirls? What kind of fibers? Collagen fibers, giving this tissue incredible strength. There are the occasional cell. Those cells are, once again, fibroblasts. But these fibroblasts are making what? Collagen, right? And it happens to be that it's laying it down in a very regular arrangement. Again, this is your tendons and ligaments. There is another one, dense irregular, that we have not asked you to identify this semester, but this is the primary tissue of your dermis. So remember up here in the inset, there's your skin, and your epidermis is up here, and your dermis is down below. Your dermis is the connective tissue on which your epithelia rests, your epidermis. So it looks more like a hodgepodge. The fibers are not as regularly arranged, right? So it's called irregular. You don't have to recognize that one. What does this one look like? Can you imagine a kind of a rubber band? Right? This is elastic connective tissue. It kind of looks like it's a rubber band that's been stretched and is recoiling. Again, you don't have to recognize it. This is going to be found, this kind of tissue is going to be found in the vocal cords. It's going to be found in the arteries and in the ligaments of the penis. Again, all places where there's a lot of stretch. The supporting connective tissues, you've already seen these, you know them and love them. So we have cartilage, and we know that the cells that make up cartilage are the chondrocytes. Bone, osteocytes. Together they create both supportive or quite strong structures. Again, 3 different types of cartilage, but all of them are similar in that they're more flexible than bone. And those chondrocytes are hanging out in spaces called what? Lacunae, right? Around the cartilage, there'll be a layer called the perichondrium. In bone, we'll see a layer called the periosteum. They're similar terms, but this is the outer layer. Think perimeter, right? Around the perimeter is the perichondrium. 3 types. Hyaline, as we'll see in a moment, means glassy. And then there's also fibrocartilage and elastic cartilage. This is the only one you need to recognize. This is hyaline cartilage. It is the most common. This is the type of cartilage you find at the ends of all of your bones where they articulate or touch each other. This is the kind of cartilage that your skeleton was once made of. Remember, we see those beautiful little chondrocytes, those eyeballs looking at us. Now, the other kind of cartilage, you don't need to recognize this, but I want you to know about it. This is the lacuna. There's the cell. Still looks like an eyeball, doesn't it? The difference is here, those chondrocytes are making a really high density collagen, making this very, very strong. Now, this kind of cartilage, this fibrocartilage, is found in your intervertebral discs. That is the cartilage between your vertebra. It's meant to be shock absorbing. It's very, very strong. So it's stronger, right? The hyaline is very weak. This is a much stronger cartilage. It's found in your knees. Any place that's weight bearing is going to have this fibrocartilage. And then finally, there is elastic cartilage. Same story. It's still eyeballs. It's still chondrocytes, but they instead make more elastic fibers. So this kind of tissue is more stretchy, and this is what you find in your ear, right, your external ear. Now remember that cartilage is avascular, which is why some people can poke holes in their ear and in their nose, and it doesn't bleed a lot because it is largely vascular free, right, no blood vessels. Bone, we'll finish up with bone here. So this is also called osseous tissue. You know that it's very strong, that there's a lot of collagen in there. It is covered by a layer called the periosteum, and it comes in 2 types. You'll see this more in lab, but there's compact, and that's the only kind we've looked at. But inside your bones, there's also spongy bone. It's a lighter bone. It's also where blood is made, and that process of blood production is called hemopoiesis. So when you're making blood, That's the process of hemopoiesis, and it happens in your spongy bones. You already know about the osteons and the lamellae and the central canal. You also already know about the canaliculi. So we see a beautiful picture here of a bone, and in the very, very center, change colors, in the very, very center, right, we have the central canal, and in there would be blood vessels and nerves traveling through there, and then around it, the entire structure from edge to edge would be a osteon, but the osteon is really more of a cylinder, so this is a cross-section through it. The cylinders are tall cylinders, and we see those layers, and each of those layers is a lamella. Now sometimes an artist's rendering is easier to see than the actual tissue. Here is an artist's rendition of the same picture, and here they have really overemphasized, but in a beautiful way. Now you can really see those canaliculi, right? Those little canals that are in between the lacunae and the osteocyte. The only connective tissue that's a fluid is blood, okay? So we'll finish up with blood, and I'll finish up with body membranes, and then very, very quickly, there's not a whole lot left here. Very, very quickly, we'll look at muscle and nervous tissue. Not much. You haven't looked at them yet. I want you to recognize them a little bit, but it won't be as much as the other ones, and I'll show you how to do that. And that will leave us just with a conversation about the skin, and that will finish up our material for the test. Now, on mastering, you will now find 3 homework assignments, one for Chapter 3, for Chapter 4, and Chapter 5. I will go through and duplicate those assignments so you can do them a second time if you choose. Do them at least once. Try to get at least 80% mastery on at least one of the 2 attempts. There's also a quiz on there. I made the quiz due not until Monday night. And I did that because, look, we're learning material on Monday, right? And in fact, I might have made it Tuesday. I made it Tuesday night. I made it Tuesday night because I had to give you time for us to finish talking about skin on Monday and have time on Monday and Tuesday to finish the quiz. Now, remember, the quiz is not timed. And you can certainly go in there now and start the quiz. You can go in there and click on the things we have talked about, skip over the things we haven't talked about, and then go back and just continue to add to what we've done. So don't wait till Monday to start the quiz. I would not recommend that because you could get caught. There's a deadline involved with that on Tuesday at midnight, and then Monday, after that is done, then you can review the quiz online for looking over things. Okay? Questions for me? Okay, well, it's hard to say, have a wonderful weekend already, but I will see you all Monday. If you have any questions, certainly contact me via email.